Hi everyone, my name is Jordao Bragantini from CZ Biohub, and I'm going to present what we are currently developing for image analysis pipeline, which at this stage we are annotating our data so we have ground truth to evaluate our algorithms. We are studying zebrafish embryos acquired with our own light sheet microscope, resulting on a huge amount of data from very big volumes at several time points. Before proceeding with the image analysis, when the data is acquired from our microscopes, it requires a lot of preprocessing, and for that we implemented our library called Dask, which uses ZAR, CuPy, and Dask to process our data in the GPU even when we need some neighborhood across different chunks of data in the spatial or time domain. For the image analysis, we want to segment and track the cell nuclei, which is very challenging in our context because the environment is very dynamic, the cells are moving a lot, leading to collisions, which undermines the performance of segmentation and tracking techniques, especially if they are not based on learning methods. To generate our ground truth data, we develop our own tool for data annotation. This tool has a main window where the user can visualize the whole data and interact with it, selecting a region to perform the annotation, which will be shown on the upper right corner, where the user can provide cues to assign labels to a subset of pixels. This subset of pixels are used to label the remaining ones, resulting on a segmentation which we can propagate across different time points. So from a single annotation, we are able to obtain segments across different frames. On the bottom right corner, we have the volume view, where we have a volume or surface rendering to evaluate how our segmentation is doing across different Z stacks. Now I'm going to present a brief demo of how the annotation works. The user selects a region for annotation. It can interact with it on the bottom view with surface or maximum projection rendering. It inserts markers providing cues for the segmentation. It obtains a segmentation, and then it can propagate across different time points, even when there is a lot of movement from different stacks. The algorithm is able to adapt to that. It also allows the annotation to occur with multiple labels, where multiple cell instances need to be annotated. Here we can see that the 3D view plays a key step, so the user is not misled from looking at only a single Z-plane. That's it for now. We hope to make this tool available to the public soon, so you can follow me on Twitter to keep up with that. And also, I would like to thank my teammates for the support and always being available for discussion, and the open source projects which we rely on, because without them, I don't think we would be able to accomplish this. Thank you, everyone.